This is Hannah Burks, and another form of technology that I found useful potentially for my classroom is CellsAlive. I selected this resource mainly because of just the raw data it has, the information it shares, the abilities it has to make it visual for students to see just the different processes and see it in action and not just, I don't know, not just still pictures, but like actually being able to visualize what happens within a cell or what happens to make like bacteria function differently and how genetics work, as well as microbes and the immune response and different cell receptors that are based on the outside of a cell. And then it also gave like study aids and quizzes and PowerPoints uh, to help in the classroom to also follow the criteria that they're teaching here in a way that should help the students benefit the most. So, the one that I looked more in depth on was mitosis, and you can start the animation, and it shows you um, the different forms of change that happens within the cycle of mitosis. And so if I click play, it'll go through and it'll bring the arrow down for each phase that's as it's going through on the graphic, which is very beneficial. And then it shows a live like example of it here. You can see more of what it looked like under a microscope. So I hit and then it goes through from interface to interface. So it's really very nice demonstration to students of like what might be going on. They can look at the PowerPoint if they need more understandings of different things. Uh, and that's also just a good review over what I've explained in class. I can show them a visual, so it's not just lecture and games, but also like visual cues that they can look back at when they're studying for tests. The standards that would be used within the interactive mitosis on Cells Alive would be ISTE 27A, which is to use technology to demonstrate students' levels of understanding of subject matter because it's allowing students to see the different phases and how it moves through the so that they can explain uh, how to how to develop these different models and understanding. The other form of standard that would be used is the ISTE 23B, which encourages the promotion of critical examination and allows curiosity. So it shows you very quickly what happens at each phase, but you can slow it down more, you can change the speed, you can speed it up, but just allows the curiosity and for them to kind of visualize it and then come up with questions that we can discuss in class based off of the cell cycle. So that was very interesting, and I could see myself using cells alive, especially in the form of mitosis and meiosis. So, have a great day.